So today we will be looking at lecture 5, which is part 1 of nervous system. And in part 1, we primarily talk about the cellular biology related to how a neuron works, how they transmit a signal, how does the signal go from one neuron to another neuron, and we sort of look at what happens if this signal transmission is disrupted. And then in lecture 6, nervous system part 2, we will be looking at the anatomy of the nervous system. So the different parts of the brain, the structure of the spinal cord, and how signal is being transmitted along the spinal cord, things like that. But today we will be focusing on just the cellular aspect of the nervous system. So it is a little bit confusing, some of the things that we have to learn in this unit. So I will be doing a little bit more overlap with the lecture recordings than I normally would, just to help you refresh the memory a little bit. We do have a quiz coming up for lecture 5, which is going to be next Monday. Right? So just like the previous unit, you will have a quiz for lecture 5, and then test number 2 will be only on 6, 7, and 8. Okay. So let's get started. Grab some papers and copy as much as you need to. So let's take a look at a typical neurons. Typical neuron. Okay, so a typical neuron looks something like this. Right, you have your cell body, and then there's going to be a long tail that comes out like this. Now, sometimes there are neurons that have 2 tails like that. Okay, so these are like bipolar neurons. Right? But to keep it simple, we'll just look at ones that have a single tail coming out. All right. So let's label the different parts of the neurons. We have little branches that are coming out from the cell body. Anybody know what they are called? If you have read about this or watch it in the recordings, what are these branches called? The ones that come out of the cell body.
Dendrites.
They are dendrites. Thank you. So these are dendrites. Okay, so dendrites, they are responsible for receiving signals. Now, these signals can be, and usually they are, from another neuron. Okay, so one neuron passes the information to the next, but it doesn't have to be. Right? It can also be from what we call a sensory structure, okay, a sensory receptor. Right? So a sensory receptor is something that would detect changes in the environment, maybe a change in temperature, maybe a change in light intensity. So when those receptors detect a change, it will then send a signal to a sensory neuron. So more on that later. Right? So right now, just know that the dendrite is going to be the receiving end of the neurons. And once again, usually it receives signal from another neuron, but it can also receive it from a sensory receptor. So receive signals from another neuron. Okay, that's like most of the case, or directly from a sensory receptor. Okay. Now, the signals that are coming in from another neuron could be what we call an excitatory signal. Okay, so we'll represent it with green arrows. Okay, so green arrows are what we call excitatory. Okay, so these are basically signals that will tell the neuron to to transmit a signal. It's like a go signal. It's tell this, it turns on the neuron if you want, okay? And it's not uncommon that at the same time, the neuron could be receiving inhibitory signals. These are essentially signals that tells the neuron to switch off, okay? So simultaneously, one neuron can be receiving multiple signals from multiple sources, some of which will be excitatory, telling it to turn on. Some of it would be inhibitory, telling it to turn off. So what does the neuron do? Does it turn on or does it turn off? Well, it's like democracy, okay? The more, the majority would win. So if there are more excitatory signals than inhibitory signals, then the neuron is going to be switched on, and then it's going to pass the signal on. If there are more inhibitory signals than excitatory signals, then the neuron is going to be turned off and the signal will not pass on, okay? Of course, that's an oversimplification of what actually happens, but keeping it simple is good. Any question about what I've said so far? All right. So this here is the cell body. Cell body. That's where you will find the usual stuff that you find in any cells. You would have a nucleus, mitochondria, ER, all that kind of good stuff, right? And down here is the axon. Axon. So signal, electrical signal, which is called an impulse, is transmitted down the axon. Okay. That only happens if there are more like green signals than red signals, right? So sometimes neurons will have insulation that surrounds the axon. So these insulations, they are called myelin, or sometimes called myelin sheath, which is called myelin. And then there are some gaps. You don't insulate the entire neuron. Okay, you have to leave some gaps for ion exchange, and these are called the nodes of Ranvier. Okay, so these are for ion exchange. Okay, the ion is like sodium and potassium, and the exchange of these ions creates an imbalance in charges, and that imbalance is what the electrical signal is. After all, electrical signal is nothing more than having electron excess and electron deficit, right? So by exchanging these ions, you create that imbalance of charges, and that signal will go down the axon. If you watch the recordings, you will know what I'm talking about. If not, that's okay. I will explain more later. Okay. Now, not all neurons are going to have myelin. All right. Some of them, especially the short one, right? They do not have myelin. And if they do not have myelin, we call them naked. Okay. It's like if we're not wearing clothes, right? We say naked, right? So naked neurons means no myelin. Okay, and they're typically shorter. Okay, usually short, usually very short axon. Okay, so the purpose of the myelin, we're getting really crowded here. Sorry. The purpose of the myelin, they are there to speed up conduction. Okay, just like the insulation around a wire, it allows the signal to go faster. Okay, and this is a kind of like a lipid type of substance. Okay, And when the signal gets to the very end, this is called the axon terminal. Okay, so the axon terminal will pass on the signal, the impulse, right, to another cell. Again, usually a neuron, but it could be something else as well. So that's your typical neuron. All right. Do they all look like this? Absolutely not. Okay. Some have, like I said, two tails. Some have special morphology, but for the most part, that's what they are. Any question before we go on? Okay. If you need to snap a picture, now is the time to do so. Okay. Let's do the pose later. Let's go back to this thing. So in our nervous system, we have two types of cells. We have the neurons. Okay, these ones they don't divide. If they die, they die. Okay? If you lose them, you don't get them back. Right? So it's very precious. We have to take care of them. We have to protect them. We have to nourish them. And that is the job of glial cells. Okay, so these support, protect, and nourish the neurons. Okay, so they can divide. Okay, and that's why brain cancer is a thing, right? Cancer is uncontrolled cell division. How do you get cancer if the neurons don't divide? Well, it's the glial cells that give rise to these brain cancers. Okay, so there are different types of glial cells, and I honestly don't remember which one we've learned, but I'm just gonna list a few for you. Okay, so we have things like oligodendrocytes. Okay, so these ones, they make myelin in the central nervous system, which is brain and spinal cord, right? And then we have something called Schwann cells. Schwann cells. And what they do is they make myelin, the stuff that speed up conduction, right? In the PNS, okay, the peripheral nervous system, which is everything else, everything else in the nervous system other than brain and spinal cord. Okay, so like you would have nerves running down your hands, right? So those nerves, that would be part of the PNS. Okay, because it's not part of the brain or the spinal cord. Okay, so the nerves that goes down the hand, the axon, they would have myelination. Okay, so the myelin for the nerves in the hand will be created by the Schwann cells. In the brain and the spinal cord goes all the way down to like, the small, the lower back, right? That is that stuff that needs to be myelinated, that will be created by the oligodendrocytes. Okay, so there are other types of glial cells, which you will learn in lecture 6, and then they do different things, and then we will summarize it once we've learned, all of them. And there are things like, astrocytes, which forms part of the BBB, right, the blood-brain barrier, things like that. You will learn. Okay. Everybody good? No questions? Okay, so this is a diagram in the study guide. I know I did this with you already in the recordings, but let's just go through it one more time. And you know, if you've drawn it already in your study guide when you watch the recordings, maybe you can draw it again on a separate piece of paper to help you, you know, consolidate the learning. So this talks about the flow of information in the nervous system. So let's say we have a hand. Okay, so we have many different types of you know, cutaneous receptors. Cutaneous means like skin, right? We have receptors that detects. Why don't you guys tell me? Okay, what kind of receptors do you think we have on the surface of our skin? What kind of things they detect? You can just type it in the chat if you want. What kind of things does your hand feel? Yep. So there's you know, heat, which is temperature, right? Pressure, pain, right? So all those things that we feel, right? Texture, all these things that we feel with our hand, not just the hand though, right? Like you can feel pain and heat with your elbow as well, right? I remember when I do the bath water for my girls when they're babies, like you test the water with the elbow, right? At least that's what my mom says, right? So you're able to feel temperature as much here in the elbow as you would in the hand. Basically, you can feel it all over your skin, right? So you have receptors for pain, for temperature, for pressure. All these receptors are what we call sensory receptors. Okay, and we have other special sensory receptors in the eyes for detecting light. We have taste bud in the tongue for detecting different tastes, right? Olfactory receptors in the nose for detecting smell, right? So the point is there is going to be some kind of sensory receptors to monitor a particular type of senses that occurs in the environment. So in this case, let's say we have a hot candle here, and we I don't know why, but like we accidentally put our hand over it without realizing it. Okay, then what's gonna happen, right? So in the hand, like I said, you have all the different types of receptors, and in this case, you would have a thermal receptor. Okay, thermal receptor. So this is a sensory receptor. They do kind of look like this for real. You just kind of have to believe me. So sensory receptor. What they do is they detect changes in the environment. Okay, now it's not possible for me to write down everything that I said, right? We will run out of space and time. So I'm going to say it one more time, right? The sensory receptors usually detects one thing only. So if I have a thermal receptors, it will be responsible for detecting change in temperature. If I have a pressure receptor, then it would be responsible for detecting changes in pressure. Okay, pressure like this, right? Like you can poke it with a blunt end of the pencil. That pressure feels different than if you poke it with a sharp end, right? Okay, so how do you distinguish the two? Well, you have a pressure receptor for that. Okay, so that's that. Now, let me put a line here to separate stuff on the left and the stuff on the right. Okay. So once the sensory receptor is stimulated, is triggered, what's going to happen is it's going to send signal to a sensory neuron. Okay, so the sensory neuron is bipolar. Okay, it has two tails. I know the recording, I actually made a mistake in the recording. I say there's many neurons, but actually it's just one really long one. So this is a sensory neuron. Sensory neuron. And it will convey that information all the way to your central nervous system. Central nervous system, or the CNS. And again, the CNS is either your brain or spinal cord, right? So that information, the information of something is hot, is going to be transmitted along the sensory neuron. Okay, so this information that is being transmitted, right? This information, the blue arrow, these are called sensory information. Right? Essentially, they are information that are detected by our senses. If this was an eyeball, right, you would be transmitting light information. Okay? If this was the ear, you will be transmitting sound information. Okay? So this pathway that takes the information from your peripheral nervous system out here, your PNS, peripheral nervous system. Sorry for the overlay. So this pathway, this is called the afferent pathway. Afferent pathway. A-F-F-E-R-E-N-T. Okay. It takes sensory information from the PNS to your CNS. Okay. So in your central nervous system, you will have another type of neuron. Okay. This one is called the interneuron. Okay, so what does the interneuron do? The interneuron will process, let me see if I can get these color code right, process sensory information and determine motor output. Okay, in other words, it takes what you have detected, and then it decides what's the best thing to do about it. So in this case, right, if you touch a burning candle, what's the sensible thing to do? Well, to remove your hand, right? So that removal of the hand, that is the motor output. Okay, so interneuron is exclusively found in the central nervous system. Right? It's only within the brain or the spinal cord. Okay, whereas the sensory neuron, the one that takes sensory information, in order to make a connection with the interneuron, it actually has to go into the central nervous system a little bit. So once you decided what is the best thing to do, then it's going to relay that information through the motor neuron. It's going to go back to the hand in this case. Okay, and then it's going to be transmitting the motor information, the motor output, right? Like this. And so this is the motor information. Okay. And this path is called the efferent. Some people pronounce it efferent. It's the same thing. So the efferent pathway will take the information from the CNS back out to the PNS. Okay. And then the hand is going to, move away. So in this case, the hand is also called the effector. Okay. Effector. Oops. The effector. The effector is basically the final destination of the information flow, the part that will be carrying out the motor output. So it could be a muscle. Right? In this case, it is a muscle. You remove your hand. It could be a gland that causes secretion of a hormone. It could be, an organ like the heart. The heart rate goes up. So that's the flow of information. Sorry, I forgot to plug in my laptop. Give me a second. All right, there we go. Any questions here? Please ask if you do. All right. So let's quickly fill this in. Did I fill this in already in the recording? Can someone please remind me? If so, maybe I won't fill it in.
Did.
I did. Okay, thank you. So I won't do that again. This is basically what I've said. Okay. And if you want the answer, you can watch the recordings again. Okay, so let's see what else is there to talk about. Okay, the division of central nervous system. Let's review that as well. Okay. Division of the nervous system. Okay, so we have the nervous system. That's like the, encompassing term. And as we mentioned already, the nervous system is separated into the PNS, peripheral nervous system, as well as the CNS, the central nervous system. Okay, so central nervous system, once again, is brain plus spinal cord. Okay. Now, the PNS is further divided into two branches. Okay. So in the PNS, we have the SNS, somatic nervous system, as well as the ANS, the autonomic nervous system. Okay. So basically, the somatic means bodily, right? So the somatic nervous system, SNS, basically has to do with all the parts of the body that you have voluntary control over. So it sends and receives, send and receives signals to and from body parts that we have voluntary control over. Voluntary control over. So like to your hands, for example, right? So if I want to move my hand, that's going to be sent through the neurons in the SNS. If I touch something hot and I feel the burn, then that's going to be related through the SNS as well, because I have voluntary control over that part of the body, right? Okay. So the sensory part, right? The sensory part, we used blue before, right? So let me try to map this out for you. Okay. So like you touch the candle, then the information is going to go all the way, all the way to your CNS. So this is the afferent path, right? Afferent pathway, okay, which is sensory. And then in the CNS, you would have the interneuron to make the decision for you. Then the information is sent back to your hand, right? That would be the efferent pathway, which would be motor. Okay. Is that okay? Like, I know it's a little bit messy, but these arrow basically helps you visualize what's going on in the previous diagram with the branches of the nervous system.
Someone, yeah,
go ahead, please.
I see the screen very small. I don't know, is it from my side or because of from your side?
Is this
because I'm unable to make it bigger, it seems.
Oh, okay. Is this a little bit better if I go like full screen? Is this
almost the same?
Is that a little bit better?
A little better, but I don't know. Usually I see it whole screen, it comes and your face comes in a small, but I don't know it's for everybody or for me.
Oh, I didn't change any settings, so
Okay, maybe something
yeah.
from my side then. Okay,
sorry
please
about
continue. Yeah.
that. Okay. Yeah, are we okay? Anyone else have any questions?
So, sir, then this somatic neuron system, the information goes to nervous system, central nervous system, right?
Yeah, so the somatic nervous system is part of the PNS, and then it will gather information, sensory information, and then send it to your CNS.
So this peripheral is different one, not involved in this AFF pathway, right?
The afferent pathway?
Yeah.
Yeah, okay. So the afferent pathway just means it's just a description of which way the information
No,
is. Yeah.
what I'm asking here, the PNS also you have in nerve system, there is two part, central nerve system and peripheral nerve system. And here the peripheral nerve system is doesn't any involvement in this SNS to CNS, right? In nerve system, in this pathway.
The PNS is not involved?
Yeah,
Is that
yeah.
okay? So it is involved through the SNS. Okay.
Oh.
Yeah. Let me finish the ANS first, and then maybe it will make more sense. Okay. I'll try to explain again, and if not, then please ask again. Okay. Now, over here, the ANS, right, autonomic system, it's basically the same thing. Sends and receives signal to and from body parts that we don't have voluntary control over. So, e.g., internal organs. Okay. Now, this part can potentially make it more confusing, but hear me out. Okay. So stop writing, everybody, and just listen. Let's say I eat too much, okay, and then my stomach is stretching too big and it hurts. Okay. How is that pain different from like me pinching myself and it hurts? So listen, right? If I pinch myself, it hurts, or like someone else pinch me, it hurts. Okay. The hurting on the hand, please tell me, is it going to be detected through the SNS or the ANS?
SNS, through the SNS.
Exactly, because I normally can control my hand, right? Okay. Now, if I eat too much and then my stomach hurts because it's too big, it's stretching beyond its normal size, that pain, is it through SNS or ANS?
That's PNS.
Well, yes, but which one, right? Is it SNS or ANS? ANS, right? Remember, PNS has two different departments, right? If you would, right? So the stomach pain would be from the ANS because I don't have voluntary control over my stomach, right? Okay. So you will also have these afferent path, right, stemming from the ANS. Okay. And then you will also have the efferent path going to the ANS as well. Okay, so both SNS and ANS, they have the receiving end. They have the afferent pathway that feeds into the CNS, and both would also have separate efferent pathway going to the different division. Okay. So there are afferent pathway and efferent pathway for all of PNS. Some goes to and comes from things that we have voluntary control over. Some goes to and comes from things that we don't have voluntary control over. Okay. Here is another example. Okay. Hopefully this will clear it up. let's say you're driving on the highway, okay? And then probably we all have this experience. Or you're driving a little bit too fast, and then you see a cop car or something, okay? Then instinctively you slow down, and then how do you feel at the same time? Like you slow down, and then how do you usually feel? Right? Like your heart rate goes up, right? Okay, and then you might get a little bit of sweaty palms. You're like, oh, I hope I didn't get a ticket, right? Okay, so what happened in that situation? Okay, listen. So you saw the cop car, right? That is afferent pathway from the SNS, right? Okay, so it goes, and then your interneuron, right, decides, oh no, I'm going too fast. I gotta slow down, right? So to slow down, do you think the motor output is gonna go through the CNS or through the ANS to slow down? Think about it. How do I tell the body to slow down? Okay, Abdullah said SNS. That's correct. Because to slow down, your leg muscle or your calf muscle has to press on the brake, right? Do you normally have voluntary control over your leg muscle? I surely hope so. So that's why it's going to go to the SNS division. And then your heart rate goes up, right? Like because of the nervousness or like scare, right? So is that going to be SNS or ANS? It's going to be ANS, right? Because you don't control your heart voluntarily, right? So the flow of information in that scenario is, it starts off with a receptor, the eye, right? It goes through the SNS of the PNS, okay? Remember, SNS is part of the PNS, right? So it goes through the SNS to the CNS, and then there are two paths coming out, okay? One goes through the SNS to your legs, slow down. One goes through the ANS to your heart to increase the heart rate. Is that better now? Who was it that asked me? Was it Ruby that asked me the question earlier? Is that more?
Yeah, thank you, sir. Yeah.
Okay, great. Thank you. Okay, moving on. Now, to make this even more confusing, is that we will have two additional division in the ANS. Okay, I'm just gonna use different colors here to highlight it. Okay, so the ANS, which deals with everything autonomy, right? Things that we don't have voluntary control over, your breathing, your heart rate, your digestive tract moving the food forward, going to the bathroom, not going to the bathroom, but like producing urine, all that stuff we don't think about, right? So all those things would fall under the ANS, and then there are going to be two subdivision under the ANS. One of them is the sympathetic, and then there is the other one, which is the parasympathetic. Okay, so the sympathetic is most active when you are in danger. Okay, most active when you are excited, stressed, or in danger, right? In danger. So Hafsa said in the chart, the fight or flight. That's exactly it. Flight or fight. Okay, so in lecture I said, if an angry dog is running towards you, you have two options. You either run away, flight, or you fight the thing, right? You fight the dog. Okay, and the sympathetic system is going to be most active during that time to help you achieve running away or fighting the adversary, right? Okay, so that would increase your heart rate, increase your breathing rate, dilate your pupils so you see more clearly, right? And at those instances, it's not time to think about digestion, right? So it will suppress the digestive system, it will suppress your urinary system and focus on diverting energy and resources for fighting or running away. Okay, so that's the sympathetic system. But that's the same kind of system that's going to be activated when you are excited. Okay, so like I don't know, like remember when you receive the acceptance letter, right, to college or to university, you get so excited, right? That euphoria, the excitement you feel, that's a sympathetic system. If you go on a roller coaster, right, that thrill, that exhilaration, that's also the sympathetic system. On the other hand, if you're just like chilling at home, not doing much, you know, getting ready for bed, very relaxed, then the parasympathetic system is going to be most active. So most active when you are calm and relaxed. Okay, so this parasympathetic system is responsible for like housekeeping items, right? Housekeeping task of the body, right? In the body. So, you know, digestion of food, elimination of waste, right? You know, cell repairing, all those kind of things. So the sympathetic and the parasympathetic system, they actually act on the same organs, but with opposing effects, right? So they act these things, right? They act on same organ or structures, but with antagonistic effect, opposing effects. Okay, so the sympathetic system would increase your heart rate, but the parasympathetic system would slow down your heart rate. Okay, because you were getting ready for bed, right? Maybe. Sympathetic system will suppress your digestive system. Parasympathetic system would promote the digestive system. All right, that's long enough time spent on these things. Any question about anything I've said so far before we move on to the really confusing part of the action potential? All right.
So this sympathetic and parasympathetic both belong to ANS, right?
That's right, ANS.
Did you, just to clarify, did you say the efferent pathway is made up of the motor neuron and carry the motor information towards the effector, right?
The efferent, the EFF?
Yes.
Yeah. Efferent is motor, afferent is sensory. Okay. Just remember, like AS spell as, right? And then the other one doesn't spell anything. I don't know if that helps. That's how I remember it.
Yeah.
Okay. Now a couple of other weird things. Just like, again, right? This is like I'm trying to like review the whole concept with you, the whole lesson with you. So it's you know, a little bit here and there, right? So the PNS, right? Like what exactly is the PNS, right? Like, you know, we talk about different divisions, but exactly what makes up the PNS, right? So we know that, again, for the, I don't know, 5th time, brain and spinal cord, right? So like this is the brain, if you're looking at it from the back. And then this is going to be the brainstem, which we'll learn next class. But we already know at least one part of the brainstem, right? We learn it in the respiratory system. Can anybody enlighten me, remind me, what's the structure in the brainstem that we learned about?
Medulla oblongata, I think.
Yes, the medulla oblongata, right? That is where you will find the respiratory center, right? Okay, so this is the brain, and then this is the brainstem, and then the brainstem will continue to come down, you know, through your entire backbone, all the way to kind of like your lower back, right? Okay, so that is the spinal cord right here. Okay, spinal cord. And then I don't know if you know this, but and we're not going to go into too much detail, but the spinal cord, once you get down to the lumbar section, okay, maybe a little bit of background information. Like, you know, your neck, right? Your neck here, this is called the cervical section. You guys know that, right? Cervical. cervical section. So you have like C1 to C7, your like your backbone, right? Your backbone is like a stack of chairs, right? And each of them has a name. So like, you know, the ones in the neck, we call them C1 to C7. And the nerves that comes out of it, we call them cervical nerves. Okay, don't worry too much about it at this moment. Just know that, you know, they have names. And then you have ones that are like in the chest area. What's another name for chest that anybody know?
Thoracic nerves.
Yeah, thoracic, exactly. So then you would have the thoracic. So that's like 1 to 12. And then, you know, you would have your lumbar. Okay, like L1 to L5. And then after that, you would have, you know, the sacral. Okay, sacral nerve. And then you would have your, I always have a hard time saying this word.
Cauda
like,
equina.
sorry, what?
I think you're trying to tell cauda equina.
Yeah. I'm trying to explain that. That's right. but you have the coccygeal nerve at the bottom. Okay. So what Mohammed is saying and what I'm trying to say is, your spinal cord actually start becoming like tail-like structures when it gets to the lumbar part. Okay. It frails out like the tail of a horse, okay, ponytail, right? And actually, that's why it's okay to stick a needle in at around, you know, L4 to L5 and take out a sample of the CNS, your cerebral, not CNS, sorry, CSF, your cerebral spinal fluid. Because when you stick the needle in, all these spinal nerve, because they're like a ponytail, they'll just move out of the way, right? You're not going to be able to poke through like the nerve, right? So why would we want to take a sample of the or like I should say, why would we want to stick a needle into your spinal cord and collect some fluid? Does anybody want to give me some examples? Someone must know here. Mothers, maybe any mothers here? Why would we stick a needle in here? Okay, so Sutena said meningitis. Yep. So for people who have potentially have meningitis and you want to verify it, then you can take a little bit of the fluid, right, from the spinal cord, and you can culture it to see if, you know, there's a bacterial meningitis or something like that. Okay. Another reason for sticking a needle into here is, you know, when a woman is giving birth, right, they could receive the epidural, right? Anybody know what an epidural is? Epidural. Yes, so epidural is basically like a numbing agent, right? That you will, yeah, it's kind of like an anesthesia. It's a numbing agent that you can inject directly into your CSF. I keep on saying it wrong. And then it will distribute throughout your spinal cord, and it will numb the pain during labor.
Yeah,
Okay.
it's painless birth. Even I had experienced this.
Yeah, I know my wife said so too. Like it helps a little bit, but then it's still not a pleasant experience. Yeah, but that's where you would insert it. Okay. And again, my point, like I'm rambling a little bit, but my point is your spinal cord becomes like a little ponytail. The name actually literally means ponytail down here, and it will move out of the way when you insert the needle. Okay, so that is the central nervous system, the brain and the spinal cord. Now, what about the PNS? Well, the PNS is going to be all these nerves that are coming out, Okay, so you have the spinal nerves, spinal nerve coming out. Okay. There are a total of 31 pairs. And from your brain, coming out directly from the brain, you would have cranial nerves. Okay, and then there will be 12 pairs. So these cranial nerves and these spinal nerves, they are all part of your PNS because they're emerging from the brain and the spinal cord, but they are not part of the brain and the spinal cord. So these are your PNS. Okay. So depending where they go to and where they come from, it might be part of your SNS or it might be part of your ANS. Do you understand? So let's say this spinal nerve, maybe it goes to your hand and it's going to be part of your SNS, right? Maybe you have one that goes to your heart, then that's going to be part of your ANS. Does that make sense? Okay. Let me see, how do I get the stuff back? I'm in full screen right now. Okay, let's do a poll. Let's do a poll. Poll. There we go. Go ahead. Oh, this is a multi-select question. I'm sorry. Wrong poll. Try again. Okay, I'll end the poll. So here, we're talking about the PNS. So PNS, we have the cranial nerves. I have the 12 pairs that comes out from the brain. We have the sympathetic system, which is part of the ANS. We have the spinal nerves as well as the parasympathetic system. Basically, anything that's not brain or spinal cord is going to be the PNS.
This spinal nerve doesn't belong to the spinal cord?
No, spinal nerve comes out from the spinal cord. So as soon as it exits the spinal cord, it's no longer part of it, right? So okay. That's why.
Then cardinal nerves means what?
Sorry, there's a cranial nerves means what?
Cranial nerves, yeah.
Yeah, cranial nerve also is part of the PNS because again, it comes out of the brain, right? So once it's out of the brain, it's no longer part of the brain, right?
So basically any nerves would be considered the PNS?
That's right. So that's a good point, actually, that I forgot to mention. Nerve, what is a nerve, right? A nerve is basically a bundle of axons in the PNS. The exact same thing in the central nervous system. So if I have a bundle of axon in the CNS, we don't call it nerves anymore. We call it tracts. Okay, that's there is a PowerPoint slides on that. Okay, so we call the same thing different names if they are found in PNS or CNS. Okay, so if it's found in PNS, we call it nerve, but if it's found in CNS, we call it tracts.
So it means CNS means just spinal cord and brain. Only two things we have to remember.
Yes, exactly.
Okay.
Okay. Now we're gonna do this in more detail next class, but you know, I'm just gonna quickly give you a little bit of foreshadowing, I guess, whatever you want to call it. Okay, so this is the brain and the spinal cord, right? Okay, so then you have the spinal nerve coming out, yes? Let's say this goes to the hand. This looks a little bit freaky. This goes to the hand. Okay, so remember we said we touch the hot surface, right? So the sensory information is gonna go this way, right? Traveling up the nerve, right? And this is gonna be PNS, right? This is a nerve, right? Sensory nerve, sensory neuron, right? And then inside the spinal cord, you will have the interneuron, and then it will come back out through the motor nerve, another one, still part of this PNS, and then we'll go back to the hand. And then, you know, to realize that you got burned, because what happens is usually like you remove your hand before you realize you got burned, right? Okay, because this is a very short path. And in order for you to realize you got burned, the signal, the sensory signal also have to go up to the brain, right? Also have to go up to the brain. Okay, so this information would be traveling along a nerve, right? Bundle of axons in the PNS. But on the way up to the brain, that sensory information will be traveling through a tract, because now you are inside the CNS, right? So that's an example of how you use these words to describe the flow of information. Okay. All right. Yeah, question, follow-up question? No? Okay. This is another one. Should be relatively easy. Go ahead. Okay, so this one I mentioned it in the recordings, but I haven't talked about it today yet. When you have one neuron communicating with another neuron, basically the signal will go from the axon terminal of one neuron to the dendrite of the next, right? Let me say that one more time. The signal will go from the axon terminal of one neuron to the dendrite of the next. So where the two kind of meets and where the two comes in close proximity, that is called the synapse. Okay, and we'll learn about what happens at the synapse later on. Okay. Next one, let's just do one more. This is very straightforward if you have been listening.
This one, there will be one correct answer or more than one?
Well, one correct answer, because it's just the CNS, right? Just the CNS.
What about astrocyte? Because I think oligodendrocyte and astrocyte both correct, right?
Astrocytes form the BBB though, not the myelin, right? The blood-brain barrier. Okay, so we have half. Basically, this is just like factual recollection, right? Oligodendrocytes is the one that forms in the CNS. Okay, this one is the PNS. So again, right, we have the same thing, pretty much the same cell doing the same thing. But if you find it in the CNS, we call it oligodendrocytes. If you find it in the PNS, then we call it Schwann cells. Okay. Anybody know of the autoimmune disease that causes the myelin to slowly degrade in the nervous system? That's right, MS. Yeah, MS. What does it stand for? Anybody?
Multiple sclerosis.
That's right, multiple sclerosis. Right, that's when your T cells attack the cells that makes the myelin. Once you lose the myelin, you lose the ability to conduct signal, and you can slowly become paralyzed, and it's something that we don't have cure for. Okay, let's see. I think we can take a break right now, and when we come back, we will do the action potential. I know 12 of you. 12 of the class watched the recordings. I assume, those 12 people are the 12 people who are here right now. So I just want to, because I don't want to waste your time, right? I want to know, would you like me to go through the action potential thing again, like here, even though, it was already covered? So in detail, if you want me to go through it in detail, please choose A. If just brief, please choose B, and then no thanks, then please choose C. Okay, the action potential with the influx, efflux. All right, so majority of the people say in detail, and some of you say no thanks. It's okay if you want to go. You could go. I will post the recording anyway, so then you can watch whatever you need to afterwards. But for the rest of the tutorial, I will go through the action potential things. Probably somewhere between brief and detail. And then we will do the application question as well. Okay, so let's take a quick break, maybe 5 minutes or so. And then when we return, we'll go through that. And if you do leave, that's okay. But the quiz is on Monday, right? Monday on this stuff. Okay, and I'll post the details about the quiz after our tutorial. I'll tell you like how many questions to expect and stuff like that. All right. So let's take 5 minutes. We will resume at 9:38. Okay, see you in a bit. Hello again. Let's keep going. So the next thing we're going to talk about is the action potential, right? And I will not go into as much detail as I did in the recording, but then I will do a little bit more than a brief summary as well. Okay, so what action potential is basically explains how the signal is passed down the axon. Okay, that's what action potential means. Okay, so a couple of things to understand, and again, I encourage you to draw this out to help you understand it. Okay, so we're gonna zoom in on the axon of a neuron. Okay, so this is the axon membrane. We have the outside, and then we have the inside. Right, so outside would be the extracellular fluid. The inside would be the cytoplasm, right? So I'll give you a moment to draw that. So along the membrane, you have various types of various types of membrane proteins, channel proteins, and we're going to be focusing on a few here. So let's put in some purple ones here. Okay, so these are the sodium potassium pump, which you learned last semester. Okay, so long story short, what the sodium potassium pump does is it will pump out three sodium out of the cell. 3 sodium out of the cell in exchange for 2 potassium into the cell. Okay? So this, of course, is active transport, so it uses ATP. Okay? Sodium potassium pump will keep on pumping sodium out and keep on pumping potassium in. Okay? So what does that achieve? Sodium potassium pump will pump 3 sodium out and 2 potassium in. So over time, what will happen to the outside relative to the inside? Is the outside going to be more positive, or is the inside going to be more positive?
The outside would be more positive.
That's right. The outside will be more positive because you're pumping more positive out than you're bringing in, right? So let's think about what she just said. It's going to be more positive outside. So the other way of saying that is that it is going to be more negative inside, right? So over time, you are going to have a negative charge on the inside relative to the outside. Okay, so positive, positive, positive. Okay, you get the idea, right? Okay. This is much easier to do on a blackboard when we're in the classroom. So what does it do? Sodium potassium pump will create a resting potential of minus 70 millivolts. Okay, so a lot to unpack here, this sentence. What is a resting potential? Okay, so let's talk about what a potential is. A potential is just a big word for charge difference. Charge difference between 2 points. Okay? If you think about a battery has a positive side and a negative side. And the battery, if you read it like those rectangle ones, it will tell you it's a 9-volt battery. Okay. If you look at the AA, right, then it will tell you it's 1.5 volts. So we measure potential difference in terms of volts. Okay. So what does that actually mean? It means between the positive terminal and the negative terminal, there is a difference of charge of 9 volts, 1.5 volts. Okay. So if we were to measure the inside of the axon compared to the outside, there is going to be a charge difference. What creates that charge difference is going to be the sodium potassium pump. How is the charge difference created? By pumping sodium out, 3 of them, and bringing 2 potassium in. Okay, so the sodium potassium pump is the one that creates, that establish the resting potential. What exactly is the how much of a difference are we talking about, right? Like the battery, it's 9 volts. So if we measure the inside and the outside, what number are we going to get? Well, you're going to get minus 70 millivolts. So there are 1,000 volts in 1,000 millivolts in 1 volt. Okay, so it's not a lot of current. We don't want to fry our neurons, but that's the number. Okay, another key word here that we have to decode is resting. Okay? When a neuron is resting, it means it is not transmitting a signal. Okay, so a resting neuron is one that is not transmitting a signal. Right? We have like millions of neurons. Not all of them are going to be transmitting signals at any given time, right? Like I'm not feeling any pain from my hand right now, so the pain, the pain nerve, so to speak, is not transmitting a signal, so it will be at rest. So when the neuron is at rest, when it's not doing anything, the charge difference on the inside compared to the outside is going to be minus 70 millivolts. Any question about what I've just talked about up to this point? Great. So let's put some identifiers here because the sodium potassium pump keeps on pumping sodium out. So on the outside, you would have high sodium. And on the inside, you would have, I'm just gonna take these out, it's kind of in my way. And then on the inside, you would have low sodium. And it's opposite for the potassium. You would have high potassium concentration on the inside and low potassium concentration on the outside. So let me ask you a very important question. Which way would the sodium wants to go if you let them do their own thing? Okay, so ignore the pump for a second. If you let the sodium move on its own, which way does it want to go? Does it want to come in or does it want to go out?
Inside?
That's right, because naturally things like to go from high concentration to low concentration, right? So if we have high sodium on the outside and low sodium on the inside, then naturally the sodium wants to come in, right? However, they cannot come in on their own, even though they really want to. That's because the cell membrane does not allow charged particle to go through, right? Okay, so even though they want to come in, they can't just yet. The opposite is true for potassium. There is high potassium on the inside, low potassium on the outside, and they really want to go out. Okay? And for the same reason, as mentioned before, they cannot do that just yet because they are charged and the membrane will not let them go through. Okay? Great. Let's add more to this diagram, which is already kind of busy. In addition to the sodium potassium pump, you have these gated channels. Okay, they are called gated channels, or sometimes they're called like voltage sensitive channels. Okay, so the red ones, they are the we'll just call them gated. Gated sodium channels. And then these are the gated potassium channels. Okay. So they're gated, meaning they can open and close like a regular gate. And what controls the opening and the closing is going to be the voltage. Right? So here we have the gated sodium channels. So, and again, right, like you just have to believe me. They will open at negative 60 millivolts, and they will close at about 30 millivolts. I rounded these numbers to make it easier for you to remember. Like if you read in the textbook and it says like 33 or something, don't freak out. Again, we just rounded. And actually there are different types of neurons as well. So we are just talking about like a typical neuron, okay? We're not talking about a neuron in the heart, for example, because those follow different rules. This is gonna sound weird coming from a teacher, but I suggest you don't go read about this on the internet, okay? Because I've tried to simplify it for you. If you read it on the internet or in a textbook, you will probably run into something that we didn't talk about and it's going to confuse you. For our purpose, we don't have to go into too much detail, okay? So just stick with what we have here. And then we have the gated potassium channels. Okay. And they will open at 30 millivolts, and then they will close at negative 90 millivolts. Think of these as like, you know, the store hours. Okay. When does Walmart open, right? It opens at like, you know, 6 a.m. and then closes at 3 p.m. That's not realistic, but you know, it's something like that. Okay. So it will open at negative 60 millivolts and it will close at 30 millivolts. So let me ask you another important question. When the neuron is at rest, when it's not doing anything, is the sodium channels open or closed? You can just think about the answer in your mind, and I'll explain in a second.
Can you repeat the question?
When the neuron is at rest, at that point, are the sodium channels open or closed?
Closed?
Yeah, because when we're at rest, the voltage is minus 70, right? And it will open at minus 60. So we are not at minus 60 yet. We're sitting at minus 70. Is not the right voltage, so it's going to be closed. Okay. Same thing for the potassium channels, right? We're sitting at minus 70. We haven't reached 30 millivolts yet, so it is going to be closed. All right, so maybe let's write that down. At rest, resting potential, both types of channels will be closed. Okay. All right. So we're gonna do the, yeah, go ahead.
The sodium potassium pump will still be on, right?
Yeah, the sodium potassium pump keeps on going. Okay, it keeps on going all the time to maintain that minus 70.
Okay.
Yeah. Okay. On another piece of paper, if you have, we're gonna do a graph. Okay. And I suggest, and please take my suggestions seriously. I suggest you draw this graph nicely when you have time on a separate piece of paper, and then stick it next to your computer when you take the quiz. Okay, it's going to help you. So big graph, okay? Like if this is your piece of paper, big graph, okay? Not one of those tiny ones, okay? Big ones. So here we go. On the y-axis, this is going to be membrane potential. So potential just means charge difference. So this is the charge difference of the membrane inside relative to outside. And then this is going to be time. Time is measured in milliseconds very quickly. Think about how quickly you can remove your hand when you touch a hot surface. That's how fast the signal is transmitted from the hand to the spinal cord and back to the hand, right? It's going to happen in less than a second, milliseconds. Membrane potential is measured in millivolts. So when we are not doing anything, when the neuron is not doing anything, it is going to be sitting at minus 70 millivolts, right? So I'm going to start my graph like this, kind of flat here, and it's not doing anything. Okay, so this is resting potential. Resting potential. Okay, help me out here, all right? At resting potential, what is the concentration of sodium on the inside? Is it high or low? Let's think about it. Okay, try not to look back. On the inside, do we have high sodium or low sodium?
Low.
Low sodium, that's right, because the sodium potassium pump keeps on pumping them out, right? And for potassium, it's going to be high. It is critical you understand all these information that we have before we move on to the next part. So I ask again, does anybody have any question? Okay, I trust you. Then a signal comes. Okay, maybe you touch a hot surface. Maybe you see some bright light when you open the window. Maybe you smell some, I don't know, mac and cheese or something. All these different changes is going to trigger a sensory receptor, right? And that sensory receptor is what will spark off a bunch of signals that will go down your sensory neurons, right? Okay, so that trigger will, let me choose a nice color for the trigger. So you have a trigger here. Okay, so this is the lightning bolt is a trigger, a stimulus. Okay, a stimulus is a change in the environment, right? So the trigger will push the membrane potential towards 0. Okay, how does it do it? I'll explain later. It won't make sense right now if I talk about it. The trigger will cause the membrane potential to become more positive. And then there is a threshold, right, at negative 60 millivolts. So negative 60 millivolts, this is called the threshold potential. Sorry, let me put it here, stimulus. Okay, so minus 60, this is called the threshold potential. Okay, so at negative 60 millivolts, do you remember what event takes place? At minus 60 millivolts, that is the time for the sodium channels to open, right? Now let me backtrack a little bit. Remember, we have so much sodium on the outside and they're just dying to come in, but they cannot because the doors are not open yet, right? But as soon as we reach minus 60, then all the sodium channels will open. And then which way will the sodium go? In or out? We talked about this before, right? In. Right? So think of it as like Black Friday. You have all these sodium, which are like the customers waiting outside, just trying to get into the store, but the doors are not open yet, okay? As soon as the doors are open, as soon as it's minus 60, then all the customers will come in. All the sodium will come into the cell. Okay, what about the potassium? Does the potassium go anywhere? They want to get out, but they can't because their doors are not open yet, right? So only the sodium will come in, but the potassium will stay where it is. Okay, so over here, this particular spot, right?
The threshold.
Sodium channels open, right? And then sodium enters the cell, right? Another $1 million question. As the sodium comes into the cell, what's going to happen to the membrane potential? Is it going to be more positive? Is it going to stay the same? Or is it going to become more negative? What do you think happens? Let's do a poll on that one, okay? Because the rest of you are very quiet. So let's take a look. My question is this. Now that the sodium channels open and all the sodiums are coming into the cell, is it going to make the inside more positive? No change for B, or more negative for C? More positive, no change, or more negative as the sodium enters the cell? And most of you said more positive, and that would be correct because the sodium is positively charged, right? So if you have all the sodium coming in, it's going to make the inside more positive, okay? So how do we show that on the graph? Well, it's going to keep on going up like this, right? Until what point? When will the sodium stop coming in? Well, They will stop coming in when the doors closes, right? And the door closes at 30 millivolts. So this is 30 millivolts, and that is when it's going to peak. Okay, so this part here, we have a name for this phase. This is called depolarization. Okay, and what's the reason for depolarization? It is sodium influx. It's just a fancy way of saying sodium coming in. Okay, depolarization. And just to fill in some knowledge gap here, polarization, that word polarization means to be more negative on the inside than outside. Okay? More negative on the inside than outside. So when you are depolarizing, you are undoing that. You're making it more positive. Okay. So what does it look like on the diagram? Okay. So I'm going to draw another one here. Go back to the other note that you were taking. I'm going to take away all the unnecessary stuff. Okay. So what just happened is this. Okay. You open the sodium channels. Okay. Remember, the axon is really, really long, right? So when the stimulus arrives, it will only open these sodium channels. The sodium channels that are further down the axon, they are still closed, right? Okay. So you have the sodium coming in, all the sodium coming in. Right? So now the inside will be more positive. Okay. However, the rest of the neuron, the rest of the axon, it is still negative on the inside, right? This part is important to understand. Okay. Any question about like what I just drawn here? So after this event, we will have high sodium, right? High sodium on the inside, low sodium on the outside. What about potassium? What should I write for potassium here? High or low for the inside?
It will still be high.
Yeah, because they didn't go anywhere, right? They didn't go anywhere. It was high before, it will still be high. And then on the outside, it will be low. Further down the axon, okay? Sodium, low or high? Low or high? What do you think?
Low.
Okay, anyone else? Hafsa also said low. Anyone else? Okay, maybe you guys are quietly agreeing, and that's good. That's low, and it's high. Why is it like that? Because the doors aren't open for them yet, right? It's very long axon. Signal hasn't get there yet. Okay, and then there would be high sodium on the inside, potassium on the inside, and low potassium. Okay, let's go back to our graph. So at this peak here, three things happen. Number one, sodium channels will close. Right? So sodium stops coming in. Right? Open and you close it, stop coming in. Number two, we are at 30 millivolts. So that is the opening voltage for potassium. Potassium channels will open. When the potassium channel opens, which way will potassium go? Into the cell or out of the cell? That's right. Potassium will leave the cell. Okay, and I'll come back to this in a second. And then the third point. Okay, I'll wait for a moment for you to copy first, and then I will tell you about the third point. Ready? Let's take a look at this picture. After the sodium comes in, right, and they will keep on pushing the voltage up, eventually you will get to 30, and then the gate will close. Once the gate closes, then the sodium won't come in anymore. Okay, and now you have high sodium here, and then low sodium on the outside. It might want to go back out, but it can't because the doors are closed. So there is only one direction that the sodium can go. Which direction is that? Look at the picture and tell me which way the sodium will go. Stephanie said down. Yeah, I think you mean down the axon, right? And that would be correct. Okay, so this is the axon. And further down, you can see it's still low sodium, right? So some of the sodium is gonna move forward. Okay, and this is very important because before the sodium moves forward, this is sitting at -70, right? So the gates are still closed. As the sodium moves forward a little bit at a time, then this number is going to go up. So it's going to go from -70 to -68, -66, -65, -61, and then finally -60, right? When it does get to -60, then the sodium channel of this section will now open, and then the sodium will come in for the next section, and then the next section, and then the next section. That's how the signal goes down. Does that make sense? Okay. So what's the third thing that's going to happen? The third thing that's going to happen is sodium channels further down the axon will open. Okay, so the sodium channels of the current section will close, but then the sodium channel further down will open. Now, after the sodium channels open, what's going to happen is the potassium, rather. After the potassium channels open, the potassium will leave the cell. As the potassium leaves the cell, is going to cause the membrane potential to come down again, right? Because you're losing positive, and it's going to come down a lot, okay, to about -90. Okay. There are two driving forces for potassium efflux leaving. Okay, number one, concentration gradient. Right? That part is easy to understand. You have a lot of potassium on the inside. You don't have a lot on the outside, so they want to go out. We knew that since the beginning. Okay, so that's going to push some of the potassium out. Secondly, another driving force is electrostatic repulsion. Okay, like charges don't like to be next to each other. Right? If you have two positive charges next to each other, they want to go as far away from each other as possible, right? So with all the sodium coming in, the inside is super positive, and then the potassium just want to get far away from each other as far as possible. So combine that with the concentration gradient, you will have a lot of sodium leaving, so much so that you are going to go down to all the way to -90, okay? And that, of course, is the point where the potassium channels will close, and then no more potassium will be able to leave. So this phase that's coming down, the falling phase, you are recreating the negative charge. So this is called repolarization. And what's causing that? Potassium efflux. Okay, efflux is just fancy word of saying leaving.
Question.
Yes, please.
So when we're at it's Colleen. You may not recognize.
Hi, Colleen.
At the action potential right to the top here. So both sodium and potassium would be high.
Yes, I was gonna do that next, but thank you. So you're correct.
Okay.
Yeah, high sodium. Why? Because they just came in. Okay. High potassium. Why? Well, because they didn't go anywhere yet, right? Okay, very good. All right, so at this low point, what's going to happen? Oh, this is so messy. Sorry. So at the low point here, right, this is where, like this point now, right? This point, this is where potassium channels close. Okay. Now, a couple of things that you know you might have overlooked. What voltage does the sodium channel open again? Just think about that in your head. What voltage? At what voltage does the sodium channel open? -60, right? -60, right? Okay. So look, we were at -70. We crossed the threshold, right? So we go up all the way up, and then it closes. And on the way down, you see we also cross -60, right? So you might be wondering, doesn't that cause the sodium channels to open again? That would be an excellent question if you were wondering that. So the answer is no, and the reason is this: these channels, when they open and then closes, after they close, they enter what's called a refractory period. So I'm just going to put it down here, and then I'm just going to explain refractory period. So during the refractory period, the channels are like resetting itself. It's like reconfiguring itself. And during that time, no matter what kind of stimulus you give it, it will not open. Okay, so the channels open and closes, then it enters refractory period, during which it's resetting itself. When it's resetting itself, it doesn't matter that you reach the triggering voltage. It will not respond. Okay, it only takes a little bit of time to reset, and then it'll be ready to go again. But it's important to have that refractory period, because if it wasn't for that, then the sodium channels would open again, and then this thing would just go up and down, like fluctuating, and that wouldn't produce a signal that will propagate forward. Okay, so if I ask you on the quiz, what is a refractory period? It is a time during which the channel is resetting itself, and it will not open, even though it reaches the opening voltage. Okay, even though you give it a stimulus, it will not respond. Does that make sense? More like when your phone dies, it needs to recharge. Yeah, kind of like that. You drain the battery, you need to recharge. Okay, but this is super fast reset. Okay, it's like another analogy is like playing dominoes with my kids. You push the thing, then you have to set it up again. Right? It doesn't matter if it falls down. If you push it again, it's not going to fall again. You need to reset it. Okay, so that's a refractory period. What's the purpose of the refractory period? It ensures the signal is going to go forward. Okay, otherwise, like I said, it's going to fluctuate up and down, and it's not going to be meaningful. Okay, so hopefully you're still with me and I haven't confused you too much. I probably confused you a little bit, but hopefully not too much. So at this point, right? What about the sodium and potassium? What should I write for the sodium? Is it high or low at this point? Let's think about it. Right? What do you think? High or low?
Sodium, low
High
potassium.
sodium. That's right, because they came in and they haven't leave yet. All right. And for potassium,
It would be low.
potassium would be low because they all just left, right? Okay. So we have two situations here, people. Two situations. Number one, we are sitting at minus 90, which is more negative than resting. Okay, so we call this point hyperpolarization. Okay, I am very sorry for the mess. The recording is probably a little bit better. Hyperpolarization. Okay, we are too negative. That's basically what it means. Hyperpolarization. So that's problem number one. We are more negative than we should be, and the cell is not going to be able to transmit another signal. Number two, we have the wrong ion distribution. Right? Remember, in order for me to go back to resting potential and the resting condition, we need to have low sodium on the inside and high potassium on the outside. So we have two problems to fix. Okay, and what do you think is going to come fix that? What's going to take all these extra sodium out and bring in more potassium and then put us back to minus 70? If you are thinking, that's right, it's the sodium potassium pump, then you'd be correct. So sodium potassium pump will recreate the resting condition so that the neuron can fire again. Okay? So we started negative on the inside. We temporarily flip to positive, and then we come back to negative again. So this temporary flip, that is called the action potential. All right. So like I said, this is a super busy graph with a lot of information. I strongly encourage you to draw this on your own and put in, information and identifiers that make sense to you, right? And then use that as like a, cheat sheet or whatever, right? For studying and for doing the quiz. Any question before we continue with this? Fantastic. Let's do some polls and see if you were attentive. Go ahead. Okay, only half of you have answered, but I'm gonna end the poll. The correct answer is influx of sodium. Depolarization is the rising phase, and it's caused by sodium coming in. Okay, these are more questions we will do in a bit. Actually, we can do this one. This one is quite difficult. Is a application question. So go ahead and try that. All right, last call. Anyone else want to answer before I end the poll? Okay, so we are tied between A and C. Does anybody want to explain, just in case you guys want to talk a little bit? People who chose A, for example, anybody want to justify the reason of why you chose A? Remember, when you choose the answers on the test, it should have a reason, right? Not like impulsive choosing, or it feels like that's the right one, right? So anybody want to justify? Yeah, go ahead, please.
I choose A because the potassium is gonna be lower. It means that if there is more supply of sodium, then automatically the potassium will be lower. I think like that, you know.
Okay, yeah, that's interesting. Thank you. Anyone else want to chime in? Okay, so I'm gonna explain. Right? Listen to the explanation and see if it makes sense. Okay, everything goes back to the chart. Everything goes back to the graph. Right? You have, that's why you have to understand the graph, right? And draw it out a couple times yourself. Okay, this is the graph, right? Okay, and this is the threshold. Right? So threshold is the opening voltage for the sodium channels. So minus 60. Okay? That's not gonna change. Okay. That has to do with the actual channel itself, right? So unless you have like a mutation in a channel or something that causes it to open and close it at different voltage, that's not gonna change. So the threshold potential will not change. That's not the answer. So I'm gonna cross that out. Okay. Now it says hypokalemia can result in hyperpolarization. So that's the first thing that this question is testing you. Do you know what hyperpolarization means? Okay, so we labeled it, right? Like this point is hyperpolarization, right? It means you are more negative than normal. So if normal is minus 70, if you have hypokalemia, you are going to be now sitting at more negative than negative 70, right? So you're going to be all the way down here. That's your new resting potential if you have hypokalemia, okay? So your resting potential will be more negative than normal, okay? And threshold potential will not change. Does that make sense so far? Well, I mean, I hope it makes sense because nobody chose B or E. So I assume you arrive at the same reasoning as I did. Okay, now next thing. The sodium channels. How do we get the sodium channels to open? Well, I have to make sure I go from this new resting potential, and I'm just gonna make up a number. Let's say negative 80 or something, all the way to negative 60, right? So in order for me to do that, the stimulus would have to be stronger. If you start off at negative 70, there's only a difference of 10 millivolts. But if you start at negative 80, now you have a difference of 20 millivolts. That is why it requires a greater than normal stimulus to trigger the action potential, to cross the threshold, right? Okay. So sodium gated channels will not open spontaneously. Okay, what does that even mean, right? Sodium channels will open spontaneously means it will open without a trigger. That is going to happen if the opposite is true. If you have hyperkalemia, okay, then maybe your resting potential is really close to the minus 60. So even though there is no strong stimulation, you might cross the threshold just on your own, and that will trigger the sodium channels to open spontaneously. Okay, if this neuron is connected to your muscle group, then that could lead to uncontrolled muscle twitching, okay? Because the gauge will open on its own, right? And then it will fire without any stimulation. Does that make sense?
Yes, but just throwing this out here. If let's say the hyperpotential was just about 75, negative 75,
Mhm.
would you still need that greater stimulus to trigger?
Yeah, because even if it's negative 75, right, you still have 5 extra millivolts to go, right, compared to normal, right? So the stimulus is still going to be greater. Does that make sense?
Yeah. Okay.
Okay. Yeah. So like this one, you might have muscle weakness. Okay. Like normally you will have to send a signal to move your hand, but now you have to send like a stronger signal in order to move your muscle or something like that. Okay. Just on an unrelated note, race horses. Right. I mean, we have a racetrack in Ontario. Never been, but I know there is one. If you want the racehorse, and this is real, like I read about it. If you want the racehorse to have stronger muscle and race faster potentially, what kind of diet would you feed them? High in potassium or low in potassium?
High potassium.
Yeah, high potassium, right? Because if you feed them high potassium, they might have high potassium in the blood, and that means they're so ready to go. They're so jittery, right? The muscle is ready to fire because the neuron is always stimulating them, and they might go a little bit faster. So that's an application question. I expect everybody who's here to get it right on the quiz. Okay. I realize a lot of the questions that I do in the tutorial, and because I can see how many people got the question right or wrong on the quiz. I must say I'm surprised, even for the ones that I take up in the tutorial, people don't get it right. So either maybe they forget about it or they haven't seen it. I don't know. But if you see this question on the quiz, I expect you to get it right. Okay. Where were we? Okay, so that is that. Let's go back to the thing here. So there's a chart. I already described it. I'm not going to fill it in again. So please make sure you fill that in on your own. And just to reiterate, it goes from resting, right? Resting potential, and then what's the next part? Threshold potential. Okay, and I should mention this as well. You sit at minus 70, like I said, and then the threshold is negative 60. Sometimes the trigger is not going to cross the threshold. Sometimes you would have a little bit of a trigger, but then it's not strong enough, so it just fall back down. Okay. Sometimes you get a little bit closer, but then it falls back down. Why is it like that? Well, because I told you, right? A single neuron could be receiving multiple inputs from multiple sources, right? So if there are more inhibitory signals coming in than excitatory signals, then you will not reach the threshold. Okay. So you must have enough green arrows coming in to push you over the threshold. If there are too many red arrows, then you won't be able to go past it. Okay? So more on that later on today. After threshold potential, what do we go to? We go to the depolarization. Depolarization. That's the sodium influx. And then you should fill these in yourself, right? And then we have the repolarization. And then we have the hyperpolarization. Okay, so you should label all these parts on the graph. You should know the reason that they are there, and you should know the relative concentration of sodium and potassium on the inside at those points. Okay. Fill in the blanks here. Let's just quickly go through this. when a neuron is at rest, the concentration of potassium is high and the concentration of sodium is low in the cytoplasm of a neuron. This resting potential is created by the sodium-potassium pump. The pump works by pumping 3 sodium out of the cell and pumping 2 potassium into the cell. And this is, again, active transport. It uses ATP. This causes the inside of the cell to be more negative than the outside. The resting potential of the neuron has a voltage of minus 70 millivolts. Action potential. Okay, so what is an action potential? It is the definition is here, so we'll highlight it later. When a signal arrives, the stimulus, it can cause the membrane potential to rise above minus 60. This is called the threshold potential. Crossing the threshold potential triggers the gated sodium channels to open. The opening of the sodium channels allows sodium to move into the cell. The influx of sodium causes the inside of the cell to become more positive than the outside, resulting in a reversal of membrane potential. This temporary reversal of membrane potential is called the action potential. So again, if you think about the graph, it is this part. That is the action potential, right? This part. It's a reversal of membrane potential because it flipped from negative to positive, and then it's temporary. So the reversal of membrane potential causes the gated sodium. Oops, I think I'm still in highlighter mode. Causes the sodium channels to close and trigger the potassium channels to open. The opening of the potassium channels will allow potassium to move out of the cell, causing the membrane potential to become negative once again. Right? So that's responsible for the repolarization phase. And because there are 2 driving forces to push this potassium out, right, the concentration gradient and the electrostatic repulsion, you're going to end up way more negative than you should be. And it's going to go all the way to minus 90. And at this point, the concentration of sodium is high and potassium is low. How do we fix the membrane potential and the ion distribution? Well, it will be restored by sodium-potassium pump. We still have a little bit more to talk about. Maybe let's just take another quick break, just help you, freshen up a little bit, and we'll come back at 10:40. If you have any question, please leave me a message. We will resume in 5 minutes. All right, let's keep going. So the last bit is going to be about the synapse. And I'm just going to do it in a short version. So you have one neuron, and then you have another neuron. Okay. So the signal is going to go from one neuron onto the next. Okay. So where they connect, right, that is called the synapse. So this is a neuronal synapse. There are other types of synapse, like between a neuron and a muscle, for example, we're going to focus on the neuronal synapse. So this here, the cell that is carrying the signal, we call that the presynaptic cell. And then over here, we have the postsynaptic cell. Okay, so the one that's going to be passing on the signal, and then this is the one that is going to be receiving the signal. Okay, maybe let's not use red because red was like inhibitory before, right? Like maybe let's just use something neutral like blue. Okay, like this. So we talked about how the signal goes down the axon, the action potential. Now we're going to talk about how the signal jumps from one neuron to the next. Okay, so if I zoom in here, we're going to have the axon terminal of the presynaptic cell. And then we're going to have the dendrite, right? Dendrite of the postsynaptic cell. Okay, let's make this big again. Okay. So at the axon terminal, you will actually have a lot of vesicles. Okay, so in the vesicles, you will have some neurotransmitters. Okay. And so these blue dots, they are neurotransmitter. Okay. So quick word about neurotransmitters. They are basically molecules, right? Molecules, chemicals, right? Molecules for synaptic communication. Okay, so next part, I'm just gonna tell you instead of writing it out, you can look at your PowerPoint slides if you want later. So some neurotransmitters are excitatory. That means it conveys like a green signal. Some neurotransmitters are inhibitory, so it conveys a red signal, right? Some neurotransmitters are excitatory in some parts of the brain, but inhibitory in another part of the brain, okay? So some are exclusively excitatory, some are exclusively inhibitory, some are location dependent. Right? Does anybody know any neurotransmitters? Things that you remember from the lecture or you read about, you heard about it. Give me examples of some neurotransmitters. Nobody know neurotransmitter? Yeah, only 6 out of 30 people watched that last part of the lecture. And this is on the last part. So examples of neurotransmitters are things like dopamine. Anybody heard of dopamine before? What's another name for dopamine? Anybody heard of that? Okay, there's a serotonin, yeah.
This is a happy hormone.
That's right, the happy hormone, dopamine, right? So if you eat a cookie and you're like, oh, that tastes so good, right? That's the dopamine creating that excitatory signal in the reward center of your brain. Okay, so that's excitatory. In another part of the brain called the substantia nigra, dopamine would have an inhibitory effect. And the purpose of that is to suppress unwanted motion when you are moving your body parts. Okay. And you know, in the lecture, I talk about Parkinson patient with low dopamine. So in the substantia nigra, and so without that suppression of the dopamine, then they shake, right? So watch the recording for a more detailed explanation. Okay, so what's the situation here? Okay, along the axon, we talked about various types of channels, right? There's the potassium channels, there's the sodium channels. And at the axon terminal, there is an other type of channels, okay? This is a calcium channel, okay? So calcium channels are normally closed, but when an action potential arrives, okay? So these are the signal, right? The sodium basically coming in. It's going to cause the enzyme to become more positive, and that will open the calcium channels. Okay. So when the calcium channels open, then the calcium will come in. The calcium will come in, and then the calcium will bind to the vesicles and push the vesicles towards the edge. Okay, so quickly recap. When the signal reaches the axon terminal, that will trigger the calcium channels to open. Calcium channels are only found in the axon terminal, not along the axon, okay? And then that will open the calcium channels, and the calcium will come in, calcium binds the vesicles, and then the vesicle will move towards the edge. Okay, so this is what's gonna look like. It's kind of like a low gray animation. So this will move towards the edge, and then it will fuse with the edge. And when it does fuse with the edge, then the neurotransmitters will be released, right, into this space between the two neurons. This space is called the synaptic cleft. Okay. Now, for simplicity, I only shown 2 neurons here, but in reality, there are hundreds of neurons around this neuron, right? So all those neurons will be exposed to the neurotransmitters. However, in order for the neurons to respond to the neurotransmitters, okay, they're exposed to it, but in order for them to respond to it, they must have the right receptor. Okay, so there's going to be a receptor here specifically designed for the binding of these blue neurotransmitters. So let's say they release pink neurotransmitters, then you would need to have a pink receptor. If you don't have that receptor, even though you are exposed to the neurotransmitters, you will not respond to it. Does that make sense? Right. So you must have the right receptor. So this receptor is, let me tell you what it is called. This is called a chemically gated sodium channel. Okay, before the stuff that we talk about in the action potential, those are voltage gated, right? Voltage sensitive sodium channels. What's the difference? They are both gated. That means they could both open and close. The voltage sensitive ones that we talked about before the break, those are controlled by voltage of the membrane. Chemically gated sodium channels, they are controlled by the presence or absence of a chemical. In this case, it would be a specific type of neurotransmitters. So to use proper, biology terminology instead of using colors, right? So like I said, right, if you have blue neurotransmitters, in order to respond, you need a blue receptor. If you have pink neurotransmitters, you need a pink receptor. So in terms of real biology terms, if you release dopamine, you would need a dopaminergic receptor. If you release serotonin, then you would need to have a serotonin receptors in order to respond. So once the neurotransmitter binds to the receptors, then the sodium channels will open, and then sodium comes in. Okay, and then what's going to happen is it will push the membrane potentials towards the threshold, and if there, I probably should have used a green signal, but whatever, right? Look, the positive signal comes in, and if there are enough sodium comes in, it will push it over the threshold, and then you would have a signal going forward. Okay, so that's basically what happens. Now you have to remove the neurotransmitters after you're done with it, right? Otherwise, you will transmit the same signal over and over again, right? Ever been to like a concert and you come back home and then you try to sleep and your ears are still ringing, right? That's because you're overstimulated at the concert with all these loud noises, right? And then it's the remnants of the stimulation still going on. So we don't want that, right? For things like pain. Okay, we touch a hot surface, you let go, and you felt the initial burn, and that's it, right? You don't want to keep on re-experiencing that over and over again, right? That's why it's important to remove the neurotransmitters. So there are two ways to remove the neurotransmitters. You can break it down, okay, with enzymes, or you can reabsorb it. Okay, so you can reabsorb it back into the vesicles where they came from, or you will have the glial cells. Remember glial cells that we mentioned at the beginning? Those supporting cells, they could help mop it up as well. Okay, so you can break it down enzymatically, or you can reabsorb them. Okay, and it's important to clear the synaptic cleft because you don't want the same signal to be transmitted over and over again. Any question? Okay. So let's do the application questions here. I will read it to you first, and we'll talk about it. Adenosine, as in like the adenosine triphosphate, the same adenosine. Adenosine acts as a neurotransmitter. When sufficient level of adenosine builds up, it slows neuronal activities and makes a person feel tired and sleepy. Caffeine is a stimulant, and its effects is related to adenosine. Based on the chemical structure shown below, explain how caffeine makes you feel energized. So, anybody want to?
Would it be that the caffeine would attach to the adenosine and more of the caffeine would be attached to the adenosine and less of the?
So you're saying that the caffeine will bind to the adenosine?
Yeah.
Okay.
Would it be?
Could be. Anyone else? All right, let's explain this. First of all, if you look at caffeine and adenosine, do you notice that there are similarities? Hopefully you will see that, okay, there is a double ring here, and then there is a double ring here, and that actually is very similar, right? Okay, so there are structural similarity between caffeine and adenosine. So let's take a look at this. Let's take a look at this. Let's say this is the neuron. Okay, you guys should be like expert in drawing neuron now. Okay. And on the neuron, you would have to have receptors for adenosine, right? In order for you to respond to it, right? Just like what we talked about. So, I'm gonna say that adenosine looks like this. I'm gonna say it's I don't know. Oops. Where were we? Adenosine is like sorry, I haven't thought this out yet. I'll just say it looks like this, okay? Adenosine looks like this. Okay, like a house. Right? So the receptor for adenosine would have to look like this, right? Does that make sense? This is like adenosine receptor. Does the picture make sense? That's the first thing.
Yep.
Yeah, okay. Thank you. So what's going to happen? As the day goes on, you build up adenosine, okay, and then the adenosine is going to bind to the receptor, okay, like this. It's going to bind to it, right? It will fit right in like this, and then the more adenosine binds to it, right, the more tired you will be, because it says in the question, when sufficient level of adenosine builds up, it slows neuronal activities, right? So all these things would tell the neuron to be like slow, slow, slow. And you're like, oh, I'm so tired. Okay. So that's the normal situation. Now, what happens if you drink coffee? Caffeine, right? So if you drink coffee, right, caffeine or Red Bull or whatever you drink, tea. So caffeine, right, it looks similar to adenosine. So I'm just going to draw a triangle. Okay? So if you flood your system with caffeine, then the caffeine will compete with the adenosine for the receptors, right? So it will bind to some of the empty receptors. It might even knock out some of the adenosine and replace them, right? And so it takes away some of the slow signal, and then you will feel energized. That's basically how caffeine works. Does that make sense? So let me just add in a little bit of things here, to help you understand the situation. Anybody drink coffee here? And if you do, or like doesn't have to be coffee, it could be like caffeinated drinks, right? Tea or whatever. If you have been a long-term drinker of coffee, something like that, can you describe to the class like what the experience is like? Like think about the first time you drink, how much you drink compared to how much you drink now. Like how would you describe that changes, or did it change at all for you?
I drank coffee once and I was falling to sleep.
You fell asleep?
Yep.
Okay, so like now or like before?
That was the first thing, because I heard coffee keeps you up because of the caffeine.
Yeah.
Because I was tired and I drank
you fell asleep.
I was still more tired. I was like really tired.
Okay,
so I never
see.
did coffee again.
Interesting. Okay, so any other long-term drinker here, long-term coffee drinker that want to explain, or no? Just tell me the answer, right? Okay. I was a long-term coffee drinker. Okay. I first started drinking coffee in does iced coffee count? Yeah, iced coffee counts. That has a caffeine in it. So I started drinking in university. Okay, and at first I just drank like a small cup, right, of coffee, and then I feel so awake. I was able to study, I don't know, like 1 to 2 a.m. in the morning, and no problem. Okay, and then I just keep on drinking since then. And then eventually I keep on drinking more and more. And by the time it was like about 10 years into drinking coffee, then I would drink a whole mug of coffee, and then I won't feel much effect. In fact, when I don't drink that mug of coffee, I feel like super sluggish. I wouldn't be able to function. I remember my coffee maker broke down one morning, and that morning I had to teach math. I had a 8:30 math class, and I was at the board, and I couldn't even do the math like as quickly as I was. It was just like so sluggish, right? And then I get these massive headache, and some of you might experience this if you stop drinking coffee out of the blue. You might have these like withdrawal symptoms, right? Okay, and so what is happening physiologically? So what happens is this: your brain actually is always trying to rebalance itself. Okay, your brain actually change throughout your life. We call this plasticity. Okay, plasticity. And your brain is always trying to recalibrate itself, trying to reset itself to regain the balance. So what happens is if you drink caffeine, it will be like, oh, okay, now that the adrenoreceptors is always occupied, I'm going to put more on it. Okay, just to regain the balance, right? Regain the balance like that. So now that you have more adenosine receptors, then the adenosine will be able to bind again, right? So now the same level of caffeine would not achieve the same effect as it once did because of these extra receptors, right? Okay, so this is called tolerance. Of course, it's not just for caffeine, right? What other things do you think your brain will do this to? Something that is perhaps more serious than caffeine. Anybody know what I'm talking about? It's on the news all the time nowadays. It was the biggest crisis before the COVID thing came.
Is it the drugs?
Yeah, what drugs? Sorry.
Weed.
Okay, weed. Yeah, but there is another one that's killing people. Actually, weed is not really killing people. Like sure, it has a lot of problems, but it's not necessarily killing people. Yes, we should say opioids, right? The fentanyl crisis, right? The opioid crisis, right? That's happening in the US as well as in Canada, right? More serious in BC, but also a problem in Ontario, right? So that's the same thing for the opioids. Your brain try to readjust when you use the opioids, and you have all these receptors. And then once you stop using the opioids without a plan, then you go into withdrawal mode, and there is like a lot of symptoms. So people just can't stop, and that actually creates the whole dependency, and the physicians no longer prescribe them. So some of the people start buying it from the street, and then those are actually contaminated with more powerful opioids in some cases, and then that causes them to become overdosed, right? So that's like the short version of what the entire problem is. There is a video that I post on eCentennial called Susan's Brain. I think you should watch that to understand more of what we are talking about. Actually, I'll post it again on the announcement at the debrief after lesson. So quickly back to this, right? So what happens is eventually after like 10 years of drinking coffee, I decided to quit. And so at first I tried to quit cold turkey, but like I told you, it gave me a lot of headaches, and I feel very tired without the coffee, right? That's because if all of a sudden you take away that coffee that you're drinking, now you have even more receptors for the adenosine to bind to, and you just feel like extra sluggish, okay? And it gives you headache, and I couldn't function. So in order for me to taper off from the coffee drinking, I actually have to reduce the dose slowly, okay? So I drink maybe half as much for 2 weeks, and then like slowly reduce the dose, and eventually I was able to quit. Okay, this doesn't happen to everybody. Like some people could just, like I know some friends who can just like not drink and not have a problem, but some people, their brain chemistry is more susceptible to these kind of things. So same thing for the opioid situation, right? If someone has been using opioid for a long time, you can't just, like people say, why can't they just quit? Well, they can't. Physiologically, they cannot do it, right? It's unbearable, the side effects. So they have to have a plan to kind of taper off. And as you slowly reduce the caffeine intake or the opioid intake, then the receptors will slowly go away, and then you will restore to a more a state that is more resemblance of what it was before you use the caffeine. I hope that makes sense. More importantly, I hope you understand how these two are related, so you can answer the application question. Okay, I ramble on and on. Let's do the next one. GABA. GABA is a neurotransmitter that causes chlorine, Cl negative, to chloride rather, to enter the neurons, or potassium to leave the neuron. Based on this information, predict the effects of GABA on neurons. When is this class ending? Soon. Okay, I promise you, probably within the next 5 minutes. Okay, so very quickly, we have the graph again. The graph. Okay, and we have the threshold here. Okay, we are sitting at minus 70 millivolts. So tell me, if you have GABA and it causes chloride to enter, if it causes Cl minus to enter the cell, you have to ask yourself, does that make you further away, like more negative or more positive compared to resting? If chlorine is coming in, And think about that for a second. Similarly, if potassium is leaving the cell as a result of GABA, is it going to be more positive or more negative? Both of these things would actually make it more negative. Okay, it's easy to understand for the chlorine, I hope. So this is negative, negative, negative on the inside. If you have more chlorine coming in, of course, it's going to be more negative, right? If you have potassium leaving, right, that means you are losing positive. If you're losing positive, you will also be more negative. So combine these two effects, GABA will actually push you further away from the resting potential. And as a result, this is going to be an inhibitory neurotransmitters. Okay. So remember earlier I said, sometimes the signal will go up, but not reach -60 and then will fall back down. That's because at any given time, the neuron is receiving both positive and negative signal. Sometimes you might have stimulation from dopamine, and at the same time, you will have suppression from GABA. So dopamine will push the threshold toward, it will push the membrane potential towards -60. At the same time, GABA will push it back down. So it's like a tug of war, right? So if you have more excitatory neurotransmitters, then eventually you will get pushed over the threshold, then you will conduct a signal. But if you have more inhibitory signal like GABA, it will keep on suppressing the membrane potential, and you will never transmit that signal. Okay. So that is that. Sorry for this like extra long tutorial. Usually we don't go this long, but there was a lot to cover. I hope you understand. And if you have a question, please feel free to email me. And the quiz, of course, is next Monday. And it will be 10% of your final grade. I will post the information online after class. So that's it. Thanks again. I will see you all next week. Bye for now.